Hi, class. In this recording, we're going to talk about some really beefy topics: excitation, contraction, coupling, and also how we can have the cross-bridge cycle. On our class website, there are some excellent A&P Flicks videos that I'm going to be hyperlinking for you. I can't put them in this recording because of copyright issues. They'd be blocked by YouTube, but I just want to make it very clear. There's some way awesome videos I'm going to be making available for you. So right now, let's dive in. This is review. We've already covered this. As we look at our muscles, skeletal muscle fibers are going to have these big characteristics. As we're looking at these characteristics, we can first have excitation. This is when we were going to have our nerve electrical signal, the action potential from our nerve, cause the muscle have an action potential or electrical signal. And then we're going to have excitation, contraction, coupling. During this phase, we'll take that electrical signal and convert it to a chemical signal so that we can start the contraction process. So we're going to take electricity in the form of that action potential and link it to those myofilaments within the myofibrils to start the contraction of the sarcomeres. Contraction itself, sometimes it's referred to as the cross-bridge cycle. I'm writing with a computer mouse right now, so that's just, I'm not going to be able to write out cross-bridge cycle in a timely fashion for you. And this is going to be a cyclical process that happens over and over and over again as the sarcomere is contracting. And then finally, we're going to talk about relaxation. After the action potential stops stimulating the muscle fiber, how do we take that contracted sarcomere and bring it back to its relaxed state? So let's go through this material. We're first going to have that action potential traveling down the motor neuron. And as it gets, that action potential gets to the axon terminal, there are going to be calcium ions entering into the axon terminal. Well, those calcium ions are entering into the axon terminal. The calcium ions bind to synaptic vesicles synaptic vesicles right there, filled with acetylcholine. After synaptic vesicles have calcium ions bind to them, those synaptic vesicles are then going to fuse with the cell membrane of the axon terminal and exositize acetylcholine. Acetylcholine will then diffuse across this space, referred to as the synaptic cleft. And as it diffuses across the space, it will bind to, in this figure, purple receptors. These purple receptors are called acetylcholine receptors. And as acetylcholine binds to the acetylcholine receptors, those acetylcholine receptors pick up two acetylcholine molecules. And when and for reference, this is on the muscle fiber. So sarcolemma means cell membrane of a muscle fiber. So as two acetylcholines bind, the acetylcholine receptor opens up and causes sodium ions to rush into the cell with the sodium concentration gradient and potassium ions to rush out of the cell with the potassium ion concentration gradient. Because this is an ion channel gate that opens up when a chemical, in this case acetylcholine, binds to it, is referred to as a ligand regulated gate, sometimes referred to as a chemically regulated gate. And this is what starts the action potential at the motor end plate of the neuromuscular junction. Just to review, the motor end plate is this region of the sarcolemma directly opposite of the axon terminal. So right there, highlighted in green, is that motor end plate. So now we're going to have the acetylcholine receptor initiating the movement of sodium and potassium ions. And as those sodium and potassium ions start moving in and out of the cell, that causes a change in voltage. And then we're going to have voltage-gated ion channels or voltage-regulated ion channels start to open and close because that initial acetylcholine receptor triggered a change in voltage. And that triggered initial change in voltage causes a downstream change in voltage, which is then going to cause additional voltage-gated channels to open and close. And this is kind of like a domino effect in that we just are going to have a wave of depolarization and then repolarization travel down the cell membrane. As that action potential or electrical signal travels across the sarcolemma, it'll get to an invagination or a transverse tubule and go down the transverse tubule because the cell membrane or the plasma membrane is continuous. As the action potential travels down the T tubule, it is going to activate voltage-regulated ion channels that are the terminal cisternae of the sarcoplasmic reticulum, SR standing for sarcoplasmic reticulum. And when those voltage-regulated channels are activated at the sarcoplasmic reticulum, calcium ions are released and then travel to the sarcomere. As those calcium ions travel across the sarcomere, those calcium ions, and now we're at the excitation contraction coupling stage. So now we're going to take those calcium ions and they'll bind to troponin. As calcium ions bind to troponin, troponin goes through a conformational shift. A conformational shift is a nice way of saying that troponin changes shape. And the troponin will go from kind of sticking out to bending over a little bit. As troponin changes shape, it's going to pull another protein, tropomyosin, which is a filamentous protein. It's going to pull that filamentous protein with it. And as it pulls tropomyosin with it, tropomyosin is moved out of the way and exposes these active sites on the actin subunits of the thin filament. By exposing those active sites on the actin, the myosin head can all of a sudden have a chemical reaction. It can interact with the active site. As we look at this myosin head, the myosin head is designed specifically to fit into the active site on the actin subunits. And what happens next is that our myosin head will be cocked. It's going to be cocked by the hydrolysis of ATP. So the myosin head will go from bent to the cocked position with ATP being hydrolyzed. This is now the contraction stage, just to emphasize this. Sometimes also referred to as the cross-bridge cycle. So step one is we cock the head or spring load the head of the myosin subunit. And then step two is that myosin subunit binds to the active site on the actin subunit. So we have the myosin-actin cross-bridge, hence the name the cross-bridge cycle. Next, the myosin head is going to bend. It's going to bend over, and as it's bending, it pulls the actin subunit past it. It'll slide past. Let me back up a slide here. So this myosin head first starts out with an oblique angle in its cocked position, and then it bends itself. It goes to an acute angle, releases ADP, an inorganic phosphate, and then in order to detach this myosin head from the actin, a brand spanking new ATP molecule needs to bind to it. And as that ATP molecule binds to the myosin head, it breaks the cross bridge between actin and myosin. This brand new ATP then is going to be hydrolyzed, and as it's hydrolyzed, the myosin head will be recocked and then can rebind to a new subunit, the next actin subunit, and then bend itself back over, and then another ATP can bind. This can happen over and over and over again. It's a cyclical process. And what it ends up happening is that one myosin head will hop from one actin subunit to the next actin subunit to the next actin subunit as actin is pulled past it. And every time a myosin head skips to the next subunit, it's going to burn up one ATP molecule per actin subunit that it advances. So that was contraction, or the cross-bridge cycle. Let's talk about relaxation. During relaxation, we're going to have that action potential that's been traveling down the motor neuron axon, or the motor nerve fiber, cease. And when that action potential stops, we're going to have calcium stop jumping into the axon bulb or axon terminal. We're going to stop exositizing acetylcholine, and then acetylcholine is no longer going to be added to that synaptic cleft. But we have a problem. We need to get rid of the acetylcholine that's already in synaptic cleft. And there's two different ways we can get rid of it. Some of the acetylcholine will spontaneously diffuse and just drift away via simple diffusion. That's a minority of the acetylcholine. We have a very special enzyme known as acetylcholine esterase. It's kind of a fat name there. I scribbled over the name. Whoops, my bad. So we have acetylcholine esterase. And this is a very special enzyme. Specifically, it breaks apart the acetylcholine molecule at an ester bond. It ends with ace, so we know it's an enzyme. And after acetylcholine esterase digests acetylcholine, the choline molecules are going to be reabsorbed into the axon terminal and recycled to make new acetylcholines. And then the acetyl functional group from the acetylcholine will then just drift away into the extracellular fluid. Let me back up a slide here. So now we no longer are going to have acetylcholine stimulating the acetylcholine receptors. As our acetylcholine receptors are no longer stimulated, we are no longer going to be triggering electrical signals on the cell. The lack of electricity on the cell means that we no longer are going to have that action potential traveling across the sarcolemma and down the T-tubule. No more action potential. So now we're going to have the sarcoplasmic reticulum burn up ATP. It will actively reabsorb calcium ions against the calcium concentration gradient by using ATP to power calcium ion pumps. Excuse me. Now as our ATP, or as our calcium ions are being reabsorbed into the sarcoplasmic reticulum, calcium ion concentration in the sarcoplasm, or cytoplasm of the muscle cell, goes down. As calcium concentrations in the sarcoplasm goes down, calcium ions start to diffuse away from troponin. Loss of calcium ions from troponin causes troponin to change back to its normal shape, and tropomyosin will slip into position the moment the myosin head detaches from the actin subunits. And once that tropomyosin slips back into position, we now are going to prevent the formation of the cross-bridge cycle so that actin and myosin can no longer interact with each other. And now our muscle stops contracting and we'll revert back to relaxation. So this is a complicated topic. It's really kind of tough to visualize with the static pictures that I showed you. I want to take a moment to highlight some truly phenomenal videos that you need to watch in order to better understand these objectives. So here's your student schedule. This has been modified to reflect the fact that, oh my gosh, everything's gone crazy with COVID-19. And now that we're 100% online, I'm going to pull up our week 7. We need to get to our week 12 or week 10 lab objectives. Let me shift to my instructor schedule. I want to show you this. Oh, of course, it's not there. So I'm just going to pull it up this way for you. You're going to have this lab objective sheet available very shortly. And you're also going to have these posted on Canvas directly for you to watch. So let's see here, weekly objective sheets. Exam 2, week 10. Thank you for bearing with me. So at the bottom of your week 10 lab objective sheet, we hyperlinked for you at the bottom of page 2, 3 phenomenal muscle contraction videos. These 3 videos do a much better job explaining what I tried to explain in this lecture recording. So please watch these videos. They're way better than what I just tried to record for you. If you have any questions, please feel free to shoot me an email or post them on class discussion boards. As always, happy studies.